Okay, good morning, everybody. I see people logging in to the webinar. We'll wait just a couple of minutes and then we'll go ahead and get started. Looks like we're going to have a good turnout today. People still coming in. Give it about one more minute and then we'll get started. Okay, it looks like the attendance is leveled off. We'll go ahead and get started. Greetings and good morning, good afternoon, or good evening, everybody, depending on where you may be logging in from around the world. I am Rob Burgess. I head up business development for Sino-Biological, and it is my pleasure and honor to welcome you all to the next installment in our Lock and Key Immunodetection webinar series, where we hear about cutting edge technologies as they apply to vaccine development, therapeutics development, and diagnostics development from a protein and antibody concentrated perspective. We have an excellent speaker today and an excellent topic today, and I will introduce our speaker here in just a minute. I did want to mention one housekeeping issue. If you don't mind, first and foremost, please log into the chat box and tell us where you're from and just say hello. We're always very interested to see where people are attending from around the world. And then second, if you have questions for the speaker, please type them into the chat box throughout the webinar. And at the completion of the webinar, I will walk through as many questions as I can verbally with the speaker. And so with that, it is also my pleasure and honor to introduce our speaker this morning. It is Dr. Christopher Locker. Dr. Locker is the CEO and the co-founder of Versatope Therapeutics, which is a clinical stage Massachusetts-based company which is focused on the development of a universal influenza type A vaccine. He previously led drug discovery projects for infectious diseases at Vertex Pharmaceuticals in Boston, inflammatory diseases at Opsana Therapeutics in Ireland, and malaria and alpha viruses vaccine development and programs at Maxigen in California. Dr. Locker completed his postdoctoral training at the University of California in San Francisco and was a Fulbright Hays Research Fellow at the Institute of Tropical Medicine in Antwerp, Belgium. He also received his doctorate in tropical medicine and his BA in biological sciences from the University of Hawaii. The title of Dr. Locker's talk today is a thermostable single-dose vaccine for pandemic preparedness. And Dr. Locker, it's an honor and privilege to have you today, and we will turn the presentation over to you.
Thank you, Rob, for your kind words of introduction. Happy to be here. So today I'll tell you about our program, our lead program, which is a universal influenza vaccine candidate. We are based in Lowell, Massachusetts. I have to dedicate this webinar to them. They did all of the work. I just led the team from behind as much as I could and get out of their way. That's always been my philosophy. So what we have is we have a phase 1 ready asset. We've engineered unique solutions for the immune system using our proprietary bacterial nanovesicles. It's been completely supported by the NIH primarily and the state of Massachusetts with non-dilutive funding. We have not had any investors to date. However, we are raising a $6 billion round to advance our universal influenza vaccine, which we call VT105. expand our nanovesicle platform for anti-inflammatories, and reach a phase 2 clinical milestone. So the opportunity is large and immediate. With the universal influenza vaccine, we see this as complementary to the current vaccine approaches. As you know, long-lasting vaccines are problematic. They have limited coverage with diverse strains. There can be a hassle with injectables or a deterrent when you have pre-existing immunity to a single strain. They can be expensive in the manufacturing process with certain types of high costs of goods and specialty reagents like cationic lipids. And there can be complications due to allergies and that kind of thing. So we've created a universal immunity solution using these nanovesicles. They are derived from the microbial membranes of gram-negative bacterial cells. And what we have to do is we have to genetically engineer them for safety primarily, as well as versatility, potency, and thermostability. So what we've done is we've captured the antigenic diversity of influenza using a very conserved and validated drug target. These vesicles can be designed with multiple functions. Primarily vaccines. There's a current product on the market called Bexsero for meningococcal B infections. They can be used for diagnostic markers, nucleic acid therapeutics, as well as drug delivery. So I'll touch a little bit primarily on the drug delivery aspect and the vaccine approach. So it is a platform opportunity. We have programs in other disease areas, including IBD, malaria, allergy, and immuno-oncology. So our solution really is to have a platform for partnering for biopharma to get these products where patients could use them most productively. We have what makes us different is we have a significant clinical impact with the ability to address multiple strains of flu, which would impress and improve economic impact. It's a strategic differentiation. It's a next generation type of vaccine, unlike messenger RNA and cationic lipid formulations. It does offer a bigger value proposition, supports global health priorities, and we believe it's a long-term sustainable solution. So why that is because with our optimized cell lines that we've developed in the lab and with Cornell University, they're engineered for very high yield productivity. They're safe. They're about 1 million-fold less pyrogenic than the wild type bacteria, so they can be injected without the nasty reactions that people typically get or at risk of septic shock. They are versatile with our gene cloning strategy. If things won't express in our system, we can simply conjugate pretty much any biomolecule. This includes small molecules as well as polypeptides and polysaccharides as well. We do have examples in each of these, and there's a plethora of publications, not only from the Cornell lab, but also from other investigators worldwide. They are high density. We do have a scalable process. They're thermostable when they're lyophilized. We have a PLGA single dose formulation, and we are clinic ready with FDA allowance for our GMP process and our clinical trial material. So, as you know, the pandemic preparedness is still broken. And every few years, the world relearns that influenza is not just an inconvenience. It's a pandemic threat. And because strain prediction fails most of the time, new vaccines have to be made every year. And these are made primarily from fertilized chicken eggs, and millions of people remain vulnerable. At Versatope, we asked a different question. What if flu prediction didn't depend on strain prediction at all? So we chose matrix 2 protein, which is an ion channel that's on the surface of the virus-infected cell. There's very few numbers on the surface of the virus itself, but there's tens of thousands of molecules on an epithelial cell that's infected with the flu virus. And what happens is it turns the epithelial cell into a virus factory. In contrast, current vaccines are focused primarily on hemagglutinin, which work great to neutralize the virus and block attachment to the host cell. However, there's multiple subtypes that recombine with neuraminidase, and it always is really difficult to predict what is going to be the next strain ahead of time to make a strain-specific vaccine. So we chose matrix 2 protein because it's relatively conserved genetically. It is market tested and clinically proven to be a validated drug target. There are 6 phylogenetic groups of the virus that cover the entire sequence space of influenza A, covering human, avian, and swine strains of influenza. When we blast searched it using GenBank, it covers almost 3,000 strains of known sequences of influenza A, and we can put all this onto a single vesicle. And we do this by having these expressed in tandem, separated by a flexible amino acid linker. So it's expressed as a single fusion polypeptide and anchored into the membrane of the nanovesicle, which is derived from the bacterial membrane, the outer membrane. We believe that this may impart lifelong or very long-term immunity. We use a strain derived from, it's called Nissl 1917. It's a common probiotic that's used for gut health. It's biosafety level 1. It's relatively safe. You can put it out on the bench, and it does not infect humans. It is very robust with our cloning strategy. We can actually get up to 2,000 copies of epitopes expressed on the surface, and thereby having this tight cluster, we can get highly amplified immune responses. No annual reformulation is needed. And again, with lyophilization, it's room temperature stable. We have a lot of data showing that it works quite well in preclinical animal model studies. So for example, with Cornell, they published this work. This was funded by the NIH. Watkins, Putnam, and coworkers showed that it actually worked a little bit better than fluvarin when you measure the viral titers in the lungs. So it's this deep infection in the lung tissues where this target actually does its job. We've also done other studies with H7N9, and we showed that, again, it reduces the viral load compared to Fluzone, which is another standard of care vaccine, which doesn't really affect these deep tissues early on in infection. Furthermore, there was a Texas variant that came out. It's a new strain. It's highly virulent in ferrets as well. This came from a human infection. You may remember that people were getting infected from cow's milk a couple of years ago. And again, VT105 outperformed Fluzone, the standard of care, as an off-the-shelf reagent to prolong life of the ferrets against a priority pathogen. Furthermore, against H5N1, against the Vietnam strain, we also saw that compared to buffer, all the ferrets had died within 7 days, but the majority of ferrets remained alive further out past 2 weeks. We also see significant reduction of viral loads in nasal washes, and we get very high endpoint titers with the VT105, which was surprising because the original variant we worked with only had 4 antigenic variants, and with the 6, we got an amplification of the immune response that seemed to be statistically significant, and it works quite well. So this formed the basis of our lead candidate, which we call VT105. Furthermore, just 2 doses is enough. If you immunize 2 or 3 times, you get about the same level of antibody titers, around 100,000 in ferrets, compared to standard of care, such as a control flu cell vaccine or buffer alone. It doesn't really induce immune responses to the target, which is matrix 2 protein. So we've done a total of 14 preclinical studies. We had some new data with these studies as well, most of which is published. These are on our website. So if you're interested in doing a deep dive on how well these work against H1N1 or H3N2 or PR8 or X47, you can feel free to check out our website, and these are all available on PubMed. So a lot of people ask, well, folks have tried matrix 2 protein, Vaccinate tried it, Sanofi tried it, Canvas tried it. There's a lot of groups out there that have tried M2 vaccine. So why are you so special? Well, we do have a strong adjuvant with this bacterial vesicle. They are expressed in tandem ectodomain repeats, which is a little bit different than other folks. Vaccinate did that, and they used flagellin, but they only had a 4X variant. And then they didn't have a bioassay either to demonstrate vaccine efficacy and potency. So we've addressed all 3 of these. We have advisors that have worked at a lot of these companies, and they have told us, I asked them the simple question, if you had to do this over, what would you do differently? And so we do have a strong adjuvant. There's a lot of good features about our adjuvants, such as pattern recognition, the way that shape of the polypeptides are expressed on the vesicles. They get trafficked to the lymphatics. They get taken up. They target the inflammasome of macrophages. We do see sustained immunity for pretty much the lifetime of a mouse. We do have other long-term studies ongoing, and they are suitable for mucosal and dermal delivery, and folks in Europe are doing these types of studies as well. So you can inject these wild type vesicles into people. They induce a nasty reaction due to the lipopolysaccharide. So we had to genetically attenuate it. In fact, what we saw was reduced cytokine signaling, primarily with interleukin 6. This has been really well profiled by the group from Siena, Italy, from the GSK team. They're really the forerunners of OMV technologies and the vaccine Bexsero. So by attenuating this TLR4 signaling, you can use them in people. With our monocyte activation test, we showed that it was 1,000,000-fold less potent than the wild type because we removed these long fatty acid carbon chains. And then what happens is it's blocked at the TLR4 receptor. It prevents dimerization, TLR4 signaling, NF-kappa B activation, and capping, if you will, on the surface of the cell membrane. So because of this reduced pyrogenicity, you can use this in people without an adjuvant. In contrast with Bexsero and other OMV technologies, even if it's 5-acyl, you have to use alum adjuvant. That's a requirement by the FDA and the EMA. So what happens when you inject these vesicles? Well, they get taken up by epithelial cells, neutrophils, and macrophages. There's an excellent reference by Tiku that talked about host immunity and cell responses to these vesicles and how they target primarily the inflammasome. And then not only that do they target macrophages, but they then trigger B cells and T cells as well to induce robust proliferative responses. Again, this is an excellent review. If you're interested on the immunological mechanisms, I highly recommend it. So what's interesting is that our fusion polypeptide is Cli-A. It's typically a toxin, but when you clone it and express it in outer membrane vesicles with the fusion protein, it doesn't have its hemolytic activity. That's attenuated as well. And we use really small doses, so we're not too concerned about it. But Cli-A, in fact, functions as an adjuvant itself. And this has been published as well by other groups. So why this is important of using Cli-A is that it forms a geometric molecular pattern on the surface of the vesicles. This is important because when your immune system sees it, pattern recognition is really key for the innate immune response. And by counting the number of pores here, if you will, as an ion channel, the configuration is an ion channel. And remember, matrix 2 protein is an ion channel. So when your immune system sees it, it recognizes an ion channel in the context of a bacterial vesicle. So what we have is the right configuration and protein shape, if you will. And that was quite a bit different than the previous matrix 2 protein approaches, which use synthetic peptides, and they tend to coil up and form alpha helices when they do that. So the Cli-A fusion protein presents 12 subunits. It is a dodecameric complex on the surface of the vesicle. And these 12 subunits, when you add single or 6 antigenic variants of matrix 2 protein, you get increasing polypeptide disorder with the increasing copies of the M2 ectodomain. In fact, this is completely correlated, and the regression is strong using a bioinformatics program. You can also visualize this matrix 2 protein. If you just have 1, it forms an alpha helix. If you have 2, that's still helical structure. 3, similarly, but by the time you get to 6, it looks like a cooked spaghetti noodle. And why is this important is because intrinsically disordered proteins lack the secondary and tertiary structures of typical peptides. So they typically won't induce robust T cells. They will induce very robust B cells because they'll bind to antibody domains. And they typically have these functions in cells where they are related to binding. And there's a whole host of intrinsically disordered proteins. It's fairly well known in vaccine design. At the bottom line is that you can get better antibody binding with greater efficiency. There's more flexibility to fit the B cell antibody receptor, the immunoglobulin, and it causes the vaccine construct overall to be more immunogenic. So this was surprising. We didn't expect that. What happens is they become more sticky. These are more enriched in polar residues. They're surface exposed, and so they lack the tertiary structured epitopes. And the hydrogen bonds and salt bridges can form per antigen, and these numbers of bonds are increased, and you get very high affinity antibody titers, coupled with the native ion channel conformation, coupled with the adjuvant properties of the vesicle, you get a very potent technology. So the third reason why the other programs didn't pass muster with the FDA and regulatory agents is because they didn't have a bioassay. So one of the first things that one of my advisors told me to do, he says, you need the bioassay. You got to make this. So we did. We typically amantadine is a known drug target that keeps people healthy. It reduces viral loads. It reduces severe disease and death. So we use this as a model to develop our matrix 2 protein, the full-length polypeptide that forms a tetrameric structure as an ion channel on the surface of the virus-infected cell. There's a couple of ways to do it. We've shown that you can actually reduce the number of plaques in a plaque assay. It is terribly labor-intensive. It can take weeks to get data, and it doesn't always work. You really need a full outfit that does this regularly, and it's terribly low throughput. And the reason why is because you have to infect the cells with the virus. You measure the number of virus-infected cells in a plaque assay. But we do have the assay developed. We have the fluorescence-based ongoing, but we tweaked it a little bit, and I'll tell you a little bit about it, about why that's important and quantitative. So the ion channel promotes viral maturation, essentially. Like I said, it's a validated drug target. It results in cellular acidification that assists in viral coat maturation and essentially turns the host cell into a virus factory and pumps out thousands of viruses. It's typically measured with a patch clamp. So you got to have a little micro needle going on the surface of the virus infected cell, and then you can measure the proton flux and the lower pH when it switches, and you can measure blocking by amantadine. This is pretty well modeled. There's other ways to do this. One way is to use GFP as a fluorescent reader, and you can measure the amantadine as well. Doing this, and what happens is when you block it with either immune serum or amantadine, the small molecule, you can reduce the switch to acidic pH, and you have this. They maintain a green color. Whereas if it's, the M2 is expressed, they lower the pH and the fluorescence is decreased. So we developed a bioassay for this. We expressed a whole bunch of variants, if you will, all 6 variants of this, including the Texas variant and a mutant variant as well of matrix 2 protein that doesn't bind to amantadine. You can sort them as well. So we sorted our clones. We developed a whole suite of clones of these expressed on mammalian cells. Spent a lot of time and energy on it, had a few people working on it. We had support from the University of Massachusetts as well with their high throughput screening. And yes, we do get dose responses. Very high concentrations of amantadine are required, but still you can visualize it in a fluorescent assay. But one of the things when we did this with our immune serum, when we used VT105, like ferret serum, for example, that came from hyperimmunized ferrets, you can see a trend there, and it is significant, but it's not always consistent. So we needed another approach. So we just went back to the basics. You know, serum often has complement in it. So we developed a complement-mediated killing assay. So it's a cytotoxic assay. So you express the matrix 2 protein on the surface. The antibody binds to the ion channel. Complement is then present in the serum as long as it's not heat inactivated. And then you can see a really nice titration down to, a 1 in 1,000. So you see a nice, really dose response with IgG. It's eliminated when it's inactivated because you have to use really high concentrations of serum. But when you have naive serum or control serum, you just don't see the activity. The current influenza vaccine does not do this. It does not have matrix 2 protein at a high enough level. It's not immunogenic enough. People with a flu infection do not develop immune responses to matrix 2 very robustly. There's a tiny bit. It's very transient. It's not immunogenic, probably because there's not a whole lot of T cell epitopes on there to induce a long-term memory response. However, when you have matrix 2 protein expressed on a bacterial vesicle, you can induce very high levels of antibodies, and the T cell is imparted by the bacterial vesicle itself. In fact, we can see very high titers across all our variants to the synthetic peptides. There was a little trick we had to do this. We had to bind them in a certain configuration, because if you just stick the synthetic peptides to a plate, they don't work that well. You have to biotinylate them, and they have to be in a certain configuration so they're upright. So you have to coat them with strep advidin, you biotinylate the peptides, and then they're in the right conformation, and then you can get a really nice ELISA titer to see these using synthetic peptides. But what about the native? People want to know, well, what about the native antigen, the full length? Well, you can measure this by flow cytometry as well. We used our GLP rabbit serum, and we find when the rabbits are hyperimmunized, they do recognize all 6 variants. It's highly genetically conserved, and with the bacterial vesicle in the right configuration, it works quite potently. Personally, I like the host cell ELISA a lot better. It just gives robust results. It's titerable. You don't have to do flow cytometry, but I like the graphics with the flow cytometry. It does have its purpose, and you can measure the number of molecules on the surface of the matrix 2 protein infected mammalian cells. So there are other folks working in OMV technologies or bacterial extracellular vesicles. We're kind of different because our platform is based on 4-acyl. We don't need any chemically detoxification of ours. We use a probiotic strain of Nissel E. coli as part of our core IP. We do have a robust pipeline in different disease areas. CLI-A is really another important aspect of this. It just assembles things properly. Not everything, but certain things. And when it has that right assembly, it works great. We've used it really well for other targets as well for vaccines, such as malaria, and in this case, matrix 2 protein. We do have the single dose formulation. I'll talk about that in a bit, and it's biosafety level 1, so you can put it out on the benchtop. A lot of these other strains are biosafety level 2, and you have to contain them. You can't just use them out on the benchtop. You need it, and that gets expensive and can be problematic. So, like I said, we're non-dilutive funded. We received millions of dollars to develop a manufacturing process through a NIAID contract that's with the National Institutes of Health and the National Institute of Allergies and Infectious Diseases. Since RFK Jr. took over, that has all changed, and our program got canceled for convenience so they could siphon off the money and put it into other programs. So what we have now is a fully defined, chemically, commercially scalable process. We have a fully chemically defined medium for VT105. Works very well. We can put it in large scale. It's a process like brewing beer. We transferred it to a GMP lab, which helped us qualify our assays. We have 24 qualified assays and 11 for information only for release, which is great. So the 40-liter scale is plenty enough material for a phase 1 and a phase 2 clinical study. You don't need a whole lot. Our doses are fairly low, and we do, like I said, a spray-dried formulation for room temperature stability. So just a snapshot of this. There's an adaptation phase to the chemically defined medium. You scale it up in the bioreactor after a couple of days, and then it goes through a downstream process following induction and an overnight growth. And you can do a buffer exchange. You can treat it with enzymes, run it over a column to get rid of the enzymes, and then it goes through a sterile filtration process and fill finish. So it's important that in the process, you monitor the bioreactor process. This took us a long time, much longer than I care to admit. And you want to get it right here at the sweet spot when they're in log phase, but not so much in log phase like at this S3, where the vesicles start to fall apart. You want intact protein and intact vesicles, and this is the sweet spot. So that's what we target when we monitor that carefully. There's plenty of critical quality attributes, as the CMC folks like to say. So a lot of it is just, size, characteristics, identity, content, pyrogenicity, contamination control. We've used all these tests. Host cell DNA, regulatory folks don't like that, so we can clean it up with nuclease. And then understanding the range and sizes, you need that tight size, if you will, for human use. One thing we found out later is that because we have a robust manufacturing process, it's relatively portable, if you will. So you can put it on a skid, and you can have a GMP process that you could locate pretty much anywhere in the world, including the developing world, for manufacturing. So that includes actually the PLGA formulation, that is polylactide co-glycolide. That actually creates a microparticle formulation. This work was published by the Cornell team. It's used for a lot of companies. There was Selecta as well here in the Boston area, and you can lyophilize it too. So this becomes a room temperature formulation. So we've done this several times. We've done several studies with it. The particles look really cool by SEM, electron microscopy. They get pretty much immunity with a single dose in a long-term infection model in a mouse. This is all published. And then also they get very high antibody titers, the same as doing 2 doses. So you don't need 3 doses or 2 doses, but this you only need 1 dose, which is convenient so you don't have to keep going back, especially if you're developing a vaccine for something like malaria, where patient compliance is challenging. People have to travel a long way to the clinic to get vaccines. So it really does open up a whole new opportunity, having a room temperature stable and single dose vaccine. And also, we've also just lyophilized it itself. We've looked at this. We get very good stability out, 12 months. This was 6 months data. So the antigen is stable after lyophilization at room temperature at 25 degrees. The protein stays intact. The vesicles stay intact. You don't need a separate formulation for V2105. You just put it in buffer. It's a little salt, and you're good to go. So it is a unique market opportunity. I don't understand net present value, but it is, I guess, pretty good because you can save a lot of lives and keep a lot of people out of the hospital. One of the things that people need to be mindful of is longevity. Influenza infects the elderly quite a bit, as well as children. So you can prevent things like heart attack, stroke, pneumonia, because a lot of the underlying causes of this are actually influenza. There's a lot of meta-analyses that are done with this. So get a flu vaccine, you'll probably live longer. So one of the things we've also taken a look at is how we can switch an allergic response to less allergic response. Cornell did this work, and it works fairly well for this because you do get robust interferon gamma type of responses, Th1, if you will, and helps shut down allergic reactions. So they've done a model with this, looking at anaphylactic scores in mice, and that with the air antigen from peanuts and with our expressing the air antigen, the glycoprotein, if you will, on the surface of the vesicles, you can express glycoproteins as well as polypeptides and just even polysaccharide epitopes as well. Cornell's done a lot of this work, works quite well. So what about immuno-oncology? I mean, so that seems to be more of an interest than infectious diseases because for some reason, a lot of people think that infectious diseases are not a problem. They are, and they will continue to be, especially if we have another pandemic. People have kind of a short memory. So what about cancer? So you can program these. You can deliver antigens for cancer. You can make a cancer vaccine. This has all been published. And the reason why is because, you can vaccinate, you can immunomodulate, you can augment like PD-1 inhibitors, that kind of thing, and making a cold tumor hot. A lot of work has been done with this. There's over 100 publications in the literature, kind of the classic basic antigen expression in B16 melanoma models, which is a pretty aggressive model in C57 mice. They used our intellectual property, which is CLIA, fused it with OVA as a fusion protein, and reduced the metastases on OVA expressing tumor cells. You can also see this with MC38, another that's a humanized cancer model, and you get survival out to 60 days, which is pretty good. You can also reduce metastases, and it's associated with a strong, robust Th1 response with interferon gamma. You can also get synergy with anti-PD-1 treatment as well, with reducing the tumor weight, both in the B16 and in the CT26 model. So it is encouraging to see that you have these different aspects of the bacterial vesicles and the OMVs. Not only do they induce robust T cell responses, but they induce NK cell proliferation as well. Doesn't seem to be much of a difference with the PD-1, but it still induces a robust innate immune response. And that includes natural killer cells, which are important in controlling metastases and tumors. So if you're interested, there's a really good review article by Shea et al. From the University of Ghent in Belgium. That study covers a lot of different aspects of OMVs and their use in augmenting anti-tumor immunity. There's tons of papers. Like I said, there's over 100 of them that have demonstrated this in multiple models. I won't go through them all, but it is a lot. But one of the things that's interesting too is you can also load the vesicles with small molecules. So this was a FLT3 inhibitor and a tyrosine kinase inhibitor that they've loaded into this, and they actually trigger anti-tumor responses as well. Reduces the tumors, and it's an interesting approach. So it's kind of like you get a 1-2 punch. You have the small molecule killing tumors, tends to gravitate towards tumors, and it reduces the tumor volume as well. So this was published as well using a proof of concept of drug loading. We've done our own work with tumor loading as well with small molecule drugs. You can see an EPR effect, which gravitates these vesicles because of the capillary structure around the tumor. And so these vesicles will just kind of migrate there by virtue of their size, because they're about 100, 150 nanometers in diameter. So they tend to clog the capillary structures, and in doing so, that surrounds the tumor. So they just gravitate towards there and they stay there. And it helps turn the cold tumor hot by activating the macrophages and switching them from a type to an M1 phenotype. You can also traffic doxorubicin as well to the tumor microenvironment. This has been shown. It works actually quite well by other groups looking at fluorescence intensity. And one of the things that's really cool, again, they used our technology using CLI-A, which is our IP, and they did a fusion protein looking at a HER2 antibody using the scFv domain. Works pretty well. You can anchor the scFv, so you don't need the full length with the Fc domain of an antibody. You can just do the antibody binding domain toward the end, and that'll fuse with the polypeptide of CLI-A, and then you can use your outer membrane vesicle for specific cell targeting. So this group did specific cell targeting. They delivered silencing RNA to cancer cells using kinesin spindle protein, and in doing so, they got very potent anti-tumor effect. This was done by Gujarati at the Korean Institute of Technology in Seoul. So we've done our own bioavailability, which is interesting. These are primarily polysaccharide in nature. There's a lot of sugar on the surface. That sugar imparts a robustness that you're not going to get with other type of liposomes or cationic lipids or chemical formulations. That robustness lends itself to lyophilization, so you can just freeze dry it, like a powder, and then add water, and then they'll just resuspend. They don't lyse in water either, which is so they're pretty robust because of the sugar. The sugar maintains that nanovesicle structure. So as a result, you can actually deliver these orally. A lot of publications have done this. You can gavage mice and show that it gets into the stomach and the intestine. You can do liquids with these. You can do pills with these. We've actually have done that work. It actually works pretty well. It'll target to the colon and to the lower GI. So if you wanted to deliver OMVs for something like colorectal cancer, you're going to the right place. They will get taken up by the GALT, or the gut-associated lymphatic tissues, and by the M cells that are present, and they will get processed and then trafficked to the other lymph nodes. They've done bioavailability studies. So it's a way to attenuate or modulate the immune system, not only just in the gut, but you can do it systemically. So if you put all this together and you think about it, have just a little bit of imagination, you can not only deliver typically insoluble drugs into the membranes of the vesicles and deliver them orally, but you could also deliver biologics. So you can clone things as a biologic, like a vaccine, and it'll get taken up by the gut. You can encapsulate it so it doesn't get completely degraded in the gut. We found that these vesicles actually penetrate extremely well. This was done by buccal and sublingual delivery of green fluorescent vesicles in pigs. The reason why they chose pigs is because pigs have an epithelial layer that's similar to humans. It's soft and tender, if you will. In contrast, rodents like rats, rabbits, and mice tend to have keratinized mouth parts, so it's kind of like the back of your fingernail. So it's kind of a tough coating, if you will. Humans don't have that. It's kind of sensitive and tender. These vesicles, because they're polysaccharide, they get taken up really fast. This was done in 5 minutes. In 5 minutes, these vesicles just penetrate. They get into the lymph nodes. They penetrate the cells, and they go right through the tissues. So this is also what happening in the gut as well. In contrast, if you have whole bacteria, they're bigger. They're 0.2 micron up to a micron in diameter. So they don't penetrate. I mean, you'll maybe a little bit, but not very much. This is the epithelial layer here. They just kind of don't really penetrate compared to the vesicles themselves. So there's a lot of different ways they can get into cells. This is pretty interesting. This is the TICU reference again. A lot of publications on it. So if you want to do a deep dive into the science, you can do that. Clathrin mediated endocytosis, caveolin mediated endocytosis, lipid RAF uptake has been demonstrated, as well as macropenocytosis and classic endocytosis. So all this has been worked out by a lot of other groups. We're not reinventing the wheel here. What this enables us to do is to deliver things to cells relatively easily, because these vesicles are biologically evolved to penetrate not only the tissues and the membranes and the epithelial layer, but also do their job once they're inside the cell and become cytosolic. So they do have a mechanism of action as well. If you deliver them orally, for example, you could deliver biologics and effectors, potent immunomodulators. These will get taken up by the cells. They will regulate the immune system as well as the microenvironment of the gut. So in this regard, they are nature's delivery tool, if you will, that works quite well. They've been biologically evolved for millions of years in people's guts and, in nature in general. So they're actually everywhere. There's, these little vesicles are doing their job. Typically in nature, what they do is they modulate their environmental niche in the gut. And in doing so, they can survive better. They can promote either homeostasis and form a microenvironment there for them to survive and communicate with other bacteria. They can deliver certain things, not like regulatory RNA and microRNA and that kind of thing. They can, antagonize other pathogens. And so when you have a healthy gut, these vesicles can help maintain a healthy gut. They also cause disease. So if you have lipopolysaccharide or a break in your tissues, these vesicles will penetrate into your body. They will activate inflammatory responses. So it's important to reach that balance where you have good gut integrity and healthy microbiota so that you don't get pathogenesis of disease. There's a really good article by Jones et al. In the Frontiers of Microbiology. I like this paper a lot. They did a lot of work on it. And they showed that these vesicles can actually penetrate the tissues. They get into the lymphatics. They go through the lamina propria. They get into the liver, so it's great for like liver diseases, as well as the lymphatics to modulate systemic organ disease. They tend to gravitate toward the small intestine, and this is what we found as well. The majority of them will stay there if you fluorescently label the bacterial vesicles following oral delivery. And what's cool about this is because there's a lot of lipid in the membranes with the outer membrane vesicles, they tend to gravitate towards the nucleus and the Golgi. So they could be, in theory, robust delivery systems for gene modulation, if you will. It may be eventually that we'll see that you can use these for genetic engineering and CRISPR delivery with enzymes if they're cloned into the outer membrane vesicles and modulate host cell function that way, and giving it by the oral route. So there's a lot of advantages, if you will, to these vesicles. You don't have to modify their chemistry on the surface. These are all done by genetic engineering. We modify that and we can tune it as well for positive cell interactions or modulate the immune system. They've been evolved by natural selection to achieve these positive cell interactions. They resist osmotic gradients as well because of their composition. Like I said, you can put them in water and they stay intact. And a lot of that is due to the polysaccharide nature of the vesicles. Size ranges are ideal really for deep penetration of tissues. A lot of other lipids, they tend to be bigger. They tend to accumulate. They tend to pop, if you will. And it's a big concern with the FDA. So in addition, there's you can give these by multiple routes. There's publications. We give them parenterally for right now, but we do have data for sublingual, buccal, oral delivery in the gut as a capsule or what have you. Intranasal delivery and transdermal delivery in patches as well. That's been shown in different melanoma models that it works quite well. They do have increased stability in serum, so that permits lower doses of them. In contrast, synthetic liposomes and chemical particles tend to be labile in serum. The half-life of exosomes typically is, about 10, 15 minutes at best. With these, it's about a half a day. It's about 12 hours. So it has a longer PK. A lot of this needs to be modeled more extensively for each one of the products that one is making to apply these. They are relatively low cost. It's inexpensive raw materials. It doesn't require mammalian cell expression, doesn't require a lot of the controls that people typically need for CMC. Manufacturing can be challenging for like 293 cells and other types of exosome therapies or even chemicals. But with our manufacturing, it's an established process, it's approved, and it is scalable. And in addition, they're lyophilized, so you can put these into a powder form, and provides different options for storage, stability, dosing, and packaging. So again, the team and myself and the board of directors, the two scientific founders are Dave Putnam and Matt DeLisa. They worked with me to launch the company. The work originally came from their lab. The management team is myself. I have a chief medical officer, chief financial officer, chief development officer that leads the regulatory work. We're joined by Drew Weissman, who is the Nobel Prize winner in vaccines and mRNA vaccines, specifically for the COVID vaccine. And then Kevin Wyant is our group leader for malaria and our Group A strep program. And Ali, our director of business operations, and Ning is now leading the lab with our inflammatory bowel disease program. We do have a robust clinical team on standby until we get funding for the program so we can advance this through the clinic. So we have biostatistics, clinical operations, quality, project management, and CMC. I have two other board members, Sam Rasty and Ed Jordan as well. They're independent and are well experienced in the biotech space here in the Boston area. The scientific advisory board is led by Bob Belshie. He is ex-St. Louis University professor emeritus, has done dozens and dozens of influenza vaccine clinical studies, and Jim Tartaglia as well. He's retired from Santa Fe, as both of these are very strong team members and have helped us get to where we need to go. So if you're interested in learning more, there's plenty of publications on our website. You're welcome to read about it. And I'm happy to take any questions. And that was my last slide. Thank you.
Great. Thank you, Dr. Locher. That was a fantastic presentation. Tons of information there. We sure do appreciate it. I want to ask our audience one more time, if you have a great question or really any question at all for Dr. Locher, please just type it into the chat box and we will get to it. While we're waiting on possible questions from the audience, I'll just kind of break the ice here. The M2 ion channel protein as a target, and I know you mentioned your IP situation briefly, I may have missed it, but can you just review that for us and tell us, do you have intellectual property or a patent issued on the M2? Do you have competitors also targeting that, or do you license the technology out to target that? Because it's very unique, and I'm just curious about it.
Well, it's a well-known target and well-established. Our intellectual property is on the VT105 vaccine itself, not necessarily on the target. Our intellectual property is the surprising indication that, or actually when we had 6 different antigenic variants, we got more robust immune responses than if we just had 4 or fewer. That was not expected. We got better survival in ferrets, and we got more robust endpoint antibody titers. So that puts our intellectual property out to 2045. It's a relatively new patent. We filed that last year. And then our core IP is around the Cly-A expression, the Nissl strain, of using that as a host strain because of its immunomodulatory properties. It produces large amounts of flagellin and has these pretty unique characteristics. It's different than other strains of E. coli or commensal bacteria. So it is a foundational IP, if you will, targeting both the composition of matter and methods of use.
Great. Fantastic. It looks like you're well positioned on that. And then the vesicle technology that Versatope has, I may have missed that as well. Please forgive me, but is that something that you would license out to other companies, or is it only exclusive for internal use? Because that's very exciting.
Yeah, well, I mean, there's a lot of folks working on it, and people have different approaches to it. Yeah, I mean, it's possible we could license it given the right circumstances.
Mhm.
Um, we tend to keep it all pretty close to our chest in terms of our strains that we've developed because they are
Mhm.
pretty unique. And that primarily the reason is because there's the endotoxin is not toxic and it's a
Mhm.
millionfold less potent than the wild type. Um, in contrast, other strains that people have done the modifications, there's multiple ways. We have, I think, 10 or 11 different mutations that we've had to do to create our 4-acyl variant because there's a lot of rescue pathways. And there's other molecules in there that tend to, you know, cause inflammation and
Mhm.
rescue, if you will, the wild phenotype. So, um, other folks, when you do 5-acyl or they do other modifications like removing a phosphate group and making it monophosphoryl lipid A, that tends to not be as potent as our strains. So
Mhm.
it's pretty unique what we've done. We have several strains. I mean, it's certainly worth a discussion if people are interested in manufacturing products to attenuate it. It is a formidable effort. It does take a long time to make these strains. We spent years making them.
Right.
The Nissel strain we have is pretty unique to us, and for now, we're going to just stick with our when you say stick to our knitting, just
Mhm.
focus on what we do best, and that's molecular cloning and process development.
Right. Sounds great. And you had also mentioned, I think, the near present value of your current flu vaccine platform is 690 million. That seems awfully low to me. I just wanted to
Oh,
comment on that. It seems to me like it would be in the multi-billion, at least annually. I mean, any more thoughts on that?
no, not really. It's all done by these mathematical financial models with people that are much more adept at doing those types of forecasting than I am. It's uh, it's kind of the cost to develop it minus the actual return on investment based on current pricing of influenza vaccines. So it's
Mhm.
been fairly consistent. There's been a couple of groups that have taken a look at this. You know, if you totally supplant the influenza market and replace the influenza vaccine with something like a matrix 2 protein for lifelong immunity,
Mhm.
it definitely has greater value for sure. You know, you're right. It's a multi-billion dollar return on the product. But when
Mhm.
you subtract all the R&D costs and you think about all the things that could go south and the probability of success, that number comes out to 690 million fairly
Mhm.
consistently. Like I said, we've had a couple of groups do these financial forecasts, these incredibly complex pivot tables and Excel files that are just
Right.
completely they're over my head. I don't know.
Yeah, over
That's not
mine
my
as
strong.
well.
Yeah.
Yeah. And just one comment, not really a question, but I saw you got Dr. Ralph Tripp on your
Yeah.
SAV, and I just love him. He and I are friends. I've licensed some technology from him at UGA many years ago. He was fantastic to work with. So I just want to say I'm glad to see you're working with him.
Yeah. Yeah. Ralph is great. Yeah, absolutely.
Yeah. Good guy. And I just wrap it up with one more question, just in case there are investors on the call. Can you remind us again about your the funding that you're seeking, your current investment status, if it's publicly available, and anything you'd like to say in that respect?
Sure. Well, we're raising a $6 million round. We have $4 million already committed, and this is to
Great.
execute our phase 1 clinical study. The $4 million that's committed is a what do they call it? A follow-on investment. We have a
Mm-hmm.
couple of groups that are interested. One is an alumni fund. The other one's kind of a local quasi VC firm here. They want to see the company succeed and they want us to grow the company.
Mm-hmm.
But we need a lead investor that we're looking for to put in that first $2 million. And that first $2 million will be the catalyst for the other $4 million.
Mm-hmm.
That's what we're looking for. We're talking to a couple of angel groups. We're talking to folks who'd probably be in the form of a safe, is our preference. So it's relatively a low bar. We have not had any investors to date in the company.
Mm-hmm.
What we'll do is with that money is execute on the phase 1 clinical study. We have our clinical team on standby. We have clinical trial sites already qualified. We're under contract with some of them. MSAs have been signed, and we have 2,000 vials of clinical trial material in the freezer allowed by the FDA. So we're ready to go. We could do additional scale up manufacturing for a phase 2 lot, further advance our partnerships and our interest in anti-inflammatories and inflammatory bowel disease as our discovery program, and develop another clinical candidate to IND ready with that $6 million raise.
Mm-hmm.
Again, this is the first seed stage investment. and positions the company for a phase one asset where some of these have actually commanded multi-billion-dollar valuations.
Yeah, that's great. There's not a doubt in my mind you will get the full round very quickly. So kudos on your solid platform and your wonderful intellectual property and technology. So with that, I think we'll wrap up. Before I forget, you see this last slide here. I just want to remind the audience that we do have a wonderful symposium scheduled in the Seattle area at the Renaissance Hotel for April 25th. It's a self-signaling breakthrough symposium. We're going to have some of the world's leading scientists and experts that are experts and very knowledgeable in self-signaling to join us. And Sino will be hosting and sponsoring that in Seattle. And I believe that's on a Saturday, April 25th. So I hope folks can make it to that. And with that, I just want to say thanks to the attendees. We had attendees really from all over the world, certainly quite a few from the United States, some from the EU, which includes Italy, and as well as folks from India. So we do appreciate y'all attending today. Dr. Loker, and forgive me for mispronouncing your name at the beginning of the intro. I do want to thank you for a fantastic presentation and taking the time to talk about Versatope and your technologies to our audience. We certainly do appreciate that. And I want to thank my colleague, Ms. Kayla Sarmiento, who just turned on her video camera. Kayla, thank you once again for flawlessly executing another immunodetection webinar. It is much appreciated. So with that, I think we will sign off and I'll say good afternoon, good morning, good evening to everybody. And thanks once again for attending. Bye-bye.